So, hello everybody attending this SciNote webinar. Today's topic is how to implement an ELN in an academic lab. And we are really delighted to have our guest speaker today with us, Kelly Lambert and Jacy Jacob. Before we introduce them, I just want to take you through the quick housekeeping. So first of all, this webinar is being recorded. So everybody that registered for the webinar will get the link to the recording. So you can also see and repeat everything that you've seen, and feel free to share it with anybody else. If you have any questions at any point during this webinar, please make sure to use the Q&A section function. You will find that at the bottom of your screen. And at any point, you can just write a question in. Both myself and the panelists will be able to see the questions. And there is a Q&A session at the end of the webinar where we'll try to address all these questions. If any remain unaddressed, we will reply directly to you over email after the webinar. And without further ado, as I said, really honored to have some of the top researchers from University of Richmond, Kelly Lambert. Kelly is a professor of behavioral neuroscience. Maybe Kelly, if you can present yourself.
Yeah, so as Clement said, my area is behavioral neuroscience, and my lab focuses on how the brain changes. We call this experience-based neuroplasticity. So I'll talk further about our lab, but I'm at a smaller liberal arts undergraduate institution, the University of Richmond.
All right, and the second panelist is Jacy Jacob. And she is a Tarwick postdoc fellow. Jacy, you want to introduce yourself?
Yeah, hello everyone. So I am a postdoctoral fellow here working with Kelly, and I exclusively sought out this experience to have a more mentor-focused undergraduate teaching and really exploratory neuroscience kind of exposure. So my PhD is in pharmacology and toxicology with a focus on a more neuropharmacology areas, substance use with opiates, alcohol, and cannabinoids. So this is kind of a jump to come and work with Kelly, but it's, you know, the brain is the brain, and pharmacology just offers tools. So I'm just using different tools and exploring more of the behavioral aspect of understanding how the brain operates. And I also have an animal science background, so I'm very comfortable with the preclinical aspect of scientific experiments.
All right, thank you so much, Jacy. So just a quick look at the agenda for today. So in the first part of the webinar, both Kelly and Jacy will have short presentations on their work and how implementing an ELN, what kind of impact it had. And then after that, we'll discuss this with both of them. We'll discuss various questions. And as the last part of today's webinar, we'll go through your questions that you submit in the Q&A session. So please, again, just be reminded, if at any point you have any questions, do share them with us. We'll definitely steer the discussion in the direction that arouses the most interest from our audience. So, okay, without further ado, Kelly, if you mind sharing your presentation.
Sure. Let's see. Right here.
Yep, that works great.
Oops, oh, that did something. Just trying to get it. There we go. Is that good?
Yep.
So yeah, it's great to be here and to share our story. I'm not sure how much of an expert we are, but we have certainly learned a lot by incorporating the use of SciNote into our lab. And I thought it would be maybe beneficial to tell you a little bit about what our lab does, to give you some context for how we're using SciNote in the lab. So as I mentioned, I'm a behavioral neuroscientist. So we're generally, our lab is interested in the brain and how it interacts with the environment and also behavior. So we've got brain, behavior, and environment. We're always thinking about how those three entities are interacting. As Jacy mentioned, we're using preclinical models. So we're using animal models to try to understand, we look at emotional resilience, how we can keep stress at bay, especially after this year. That seems to be very relevant. So we're also looking at different ways, training and how parenting, other aspects changes the brain. So most of my work has been done on this rat. The rat has a small brain, only about 2 grams compared to our perhaps 1,400 gram brain, but mine is probably considerably less, lighter after this year. So it is a smaller model of our brain, but it has pretty much all the same parts and neurochemicals and hormones that influence it. So, and the cellular types and profiles. So it's a good place to start. And this animal is a survivor and very flexible. And so I learn a lot from these preclinical models. I think they're very complex. We try to not, especially as I get older, to think about the limitations of only looking at rodents. And a lot of times people in my field, we like to look at mice and rats. We're also branching out to looking, and we're interested in the clever raccoon. So that's a different laboratory experience. We can't keep them in the lab. So we're collaborating in different ways to look at that wild species. We do some non-invasive work with primates at a wonderful facility, Monkey Jungle in Miami, Florida, here in the States. Looking at, I just got back the end of last week looking at these Java macaques. And some squirrel monkeys have wonderful parenting behavior. We have looked at a mouse model, the California deer mouse, looking at paternal behavior, and our wonderful Long Evans laboratory rats. And recently we've gotten into wild rats, which is a whole different story too. So we have a lot going on in our animal kind of profile. And just to, maybe make you smile, we also do some thinking of neuroplasticity. You saw the driving rats in the news. I was the crazy professor behind that. Looking at how if we can look at something that is training, long-term training, a month or more, or several months, especially teaching them to steer. But so often we use very simple tasks, and those may not translate to humans where we learn very complex tasks. So we're looking at the effects of environment and such on even driving in rats. So to let you know a little bit about, more about our context and some of the challenges I face as an undergraduate professor doing research with students. As I mentioned, I'm at the University of Richmond. We have about 3,000 undergraduate students. I am the only behavioral neuroscientist here. We do have other neuroscientists. So that's a challenge, and well, it's a good challenge. It forces me to collaborate outside of the university. With undergraduate students, I see Jacy here and a research tech, but these are undergraduate students, another postdoc here. So I have a good fortune in the last decade or so to have a postdoctoral fellow to help me. But one of the challenges with undergraduate students is that they have a high turnover rate. So that study they did one summer, and we were very careful about making sure everything's documented. And it was in that special notebook with everything, with the little markers and such. Even though I try to get things written up while that student is still at the university, maybe the next academic year, maybe I'm sharing too much, or maybe you're more organized than I am, but it's usually another year or so and the student has already graduated, and I'm writing up the methods and I'm looking to find that notebook and I'm emailing them. They're already in medical school. Do you remember where you put the notebook? We knew that we said that we weren't going to forget this. And so that's been a struggle. The undergrad, I love working with undergraduates and all the new ideas that they bring. But that has been a struggle trying to keep track of their research. And I try to put every student on original research so that if we're using the animals, we're contributing to the literature. So finding those notebooks, keeping them organized has been a challenge through the years. And I'm looking at, I've been doing this for 3 decades now, so it has been a struggle. I have this graphical abstract of an article that we currently have in press of our raccoons, which is new, just as a prompt to let me talk about, or prompt me to talk about our collaborative research. Something like a raccoon, as I mentioned, we can't keep them in the lab. So we're collaborating with the United States Department of Agriculture, with wildlife ecologists. This article, I don't know if it's good or bad, but represents about 10 years of work with 21 colleagues and students at 9 institutions. So when you're thinking about keeping track of the information over that time span with that many individuals, students coming in and out, and now this is at 2 different institutions. I moved about 4 years ago myself. So another challenge, how do we keep track of all this information? So I was just going along struggling with that. And then COVID hit, and it gave us a little bit of time to reflect on how can we use this time to maybe reassess what we're doing, and how can I be more efficient and effective with the research and our supervision with the students. So I'd heard about these electronic lab notebooks, and I just kind of Googled and looked around, and I did read a very favorable review of SciNote and contacted them to start to talk about what, and I thought that maybe we were too small of a lab to benefit from an ELN, but as I described what our challenges were, it seemed like it would be a good fit. So we had some initial conversations with BID, and then talked to our lab team members who were still we were having Zoom meetings every other day or such. During the time that we were off, we were able to get back to the lab pretty quickly. But everyone was in agreement that this was worth worth trying because we wanted to do better. So we had some weekly training sessions, and to see how we could organize this. And Jacy really did the heavy lifting and organizing. She's going to tell you more about that, but I'm kind of the idea person to see if this would be a good fit. Value added, as we were going through this process and really thinking about our different projects, the way this is organized in experiments, their inventories, what our vendors were, our protocols, it really is great to bring in. I saw a learning opportunity for students that we could share this process with them, and we're still getting up and running with this. We've had it for about a year, but the students are just now getting back into the lab. But as we identify our projects and questions and materials and such, and Jacy is going to, she will give you a better example of that. But I love how we were forced to conceptualize everything that was going on in our lab in a way that I really hadn't done before. I also serve on review panels for National Institutes of Health here in the US, and have started to think that and incorporate this in my applications, because I know that with professors working with undergraduate students, a concern of the reviewers is, how are you going to do this with that turnover? So this is one of those responses. I've thought about this, because you always want to try to be a step or two ahead, and using an ELN will help us coordinate and keep everything organized and reliable, so that we can finish this project and do it accurately. This is a book that I read. I've been a little bit obsessed with it lately. All the students in my lab, all 7 of them, and our trainees, supervisors are reading this book this summer, Science Fictions. And this is by Stuart Ritchie. And it talks about some of the flaws and biases with misinformation, but focusing on science. And as I was reading this, I was going kind of like, oh, maybe, hopefully we're not doing this. We need to look at this more closely. It's made me more vigilant of the incentives that are used in our system, how easy it is to kind of go down a path of being too focused on maybe citations and getting those positive results out. And it's a good reminder that, you know, at the end of the day, we just want the accurate answer to the question that we're asking. But I think this is great to remind us how to do science in this most accurate and effective way possible. I wanted to mention this because I'm hoping that SciNote will keep me from being in one of his books in a chapter, maybe something I missed or a photo, you know, photomicrographs that weren't accurate or something. Hopefully we're not doing this, but it made me feel better that we're a little bit more accountable with an ELN. And I can track things and have that transparency. So it makes me feel a little bit better that I may not be included in a future book that Stuart Ritchie has written. In fact, he's going to meet, he's very nice. He's going to do a Zoom session with my students and some other students and labs at the University of Richmond, because I think as scientists, we all need to be out front with this and doing as much as we can to enhance the integrity of the science and not just the number of publications that we have. So that's a little bit about our lab and what brought me to SciNote. And I will stop sharing. And Jacy's going to show you kind of a case study of how we have used it.
All right, thank you so much, Kelly, for this. Just a couple of questions before we hand it over to Jacy. So maybe just to clarify, like how did you know, COVID era and all the pandemic and the lockdown, how did that impact your work? Like, did you guys go home? Were you doing any work during this time?
Right. So the students were all sent home, and we were sent home until I think at the beginning of June, Jacy, or first week of June last year, we were able to come into the lab. We maintained our animals. We had a schedule, a plan, so that we were all rotating, going in, taking care of the animals individually. And because, again, we're a small scale. It's not like a larger university where that wouldn't have been possible. So we maintained our animals, and then we worked during COVID when we were not able to work in the lab. We had maybe 2 or 3 Zoom sessions a week. We shifted to writing and then thinking about how we could do things more effectively, and then came back and started to do, we didn't have the students to help, but did what we could do to try not to get too far behind on our grants. So I think we used our time wisely, but I'm sure everyone used a little bit of that time to reflect on how we're using our time, and being forced to do something different is great for the brain. Someone studying neuroplasticity, it gets us out of the same lane and we're thinking and seeing things differently. So we're all trying to look at something positive of a very traumatic experience, but changing our perspectives has been one positive output or a result of COVID.
I couldn't agree with you more. Like we see here at SciNote, we see a lot more interest in ELN and digitalization in general, which I think is a great, let's say, I mean, one of the good things coming out of COVID. Yeah. And I'll have a bunch of comments and questions on this, but maybe we save those for the discussion. So Jacy, maybe if you can try sharing your screen.
Sure, absolutely. So I'm going to go live here and share our actual SciNote page. If you're already a user, this should look familiar, but if you're not, this is the overview page. So this is just where any of the users that are assigned to a specific task will see what, I guess, elements of the projects need attention. There's a nice little calendar. as well as the recent work. So if you are in a position like myself, where you're one of the account administrators, as well as serving in some sort of lab manager role, it's also really nice to be able to track what the recent work on your SciNote account has been, to ensure that just at a broad overview or a quick glance, you can see the productivity of your lab. But honestly, even though we start on the overview page, I spend the most amount of time in the projects area. So here you can see where we've organized our projects. And as I've mentioned before to Clement and others at SciNote, one of my favorite updates is this incorporation of folder organization. So you heard Kelly mention our driving rats, and we've had multiple cohorts of these animals now. So I've actually separated and distinguished a folder for each of those cohorts of driving rats. You also heard that we work with animals that are not traditionally utilized in neuroscience research, one of those being the raccoons. And so just to give you a quick glance, when I open our raccoon folder here, you can see our various raccoon studies. In fact, this is the one that was just published in the Journal of Comparative Neurology. And so as I go through my different layers, you can see I'm getting closer and closer to looking at the actual project details. So just for cohesive and generalizable information, I wanted to start with our grant project that's funded through the National Institutes of Mental Health. And this is another project that I thought was worth highlighting for you all, because it's not a single user who's working on this project. I don't know many cases where there's a single person, but maybe a graduate student who's got most of the ownership of a project. But here, especially with our undergrads, and as well as having a postbac in the lab and a research technician, it's a really collaborative environment, not only with other institutions, but within our lab itself. And so I just wanted to utilize this example because it's such an all hands on deck example. So most grants, as you know, are typically split into different specific aims. And so even though I have one project being this grant project, I have different sub-projects or experiments that are named based on the specific aim in which they are encompassing. And so specific aim 1 here is one of our first forays, I guess, into kind of adapting SciNote into our lab and the way that we work. And this did take some time to really think about how best to adapt the way that the grant was written and the way that we typically do our science at the bench into a digital format that is capable of being understood by not only the users that are creating this task flow, but those that are going to come back and be able to follow what we did in the future. So one of the things that the postbac, Emily here and I settled on was the fact that we need kind of a home base or kind of a central point where the entire web is going to be created from. And so what we've created here is that every single experiment or project that we use starts with animal information, because that is a constant across nearly all of our experiments. We will always have either rats in the lab that we're working on behavior and then follow up immunohistochemistry or other tissue studies, or we'll receive brain tissue from other collaborating labs. So it all applies. So we always start with this animal information tab. And let me just go scale down just a second so you guys can actually see this. So when we're creating our web, one of the things that we really spent a lot of time working through and kind of customizing for our needs was the fact that we didn't have streamlined, or what I would say more linear processes, where you start with a single step, something like a qPCR experiment or a Western blot or an ELISA, where it's very linear, where there's clearly a step 1, 2, 3, 4, etc. You'll notice, again, if you're a user of SciNote, that a lot of the demos, and fairly so, for clarity, they are very linear processes with these webs. However, our lab, and I know a number of other biomedical laboratories and other neuroscience focused laboratories that utilize preclinical models, aren't just looking at the tissue side, you're also looking at behavior. And so this is where we've really created this ability to kind of branch and create a non-linear task flow. So because we have our central hub here of our animal information, and I'll go into those in just a couple minutes to show you a little more detail, we branch all of our behavior in one direction, which then becomes linear because they are in a systematic flow. And then we can create other branches from that original starting point that target our endpoint analyses, whether that be immunohistochemistry or other wet lab experiments that we're doing in an ex vivo format. So you can see here, this experiment and this project starts with behavioral training, and then we're interested obviously in how that behavior is affecting how the brain is operating and changing between a treatment group and a control group. And so then we're utilizing aspects like neurochemical changes through immunohistochemistry targets. So here a very common BDNF target, double cortin. Those are antibodies that we use often in the lab, as well as looking at a neuroendocrine glance. So we're looking at corticosterone and DHEA as 2 hormones of interest, and those are ELISA kits. And so then the really nice thing that Emily, actually, our postbac, came up with was, even though within each of these tasks there are places where you can upload protocols and upload results, just to keep everything streamlined and a quick way to see overview results, we always end our task flow with a final results section. So it's kind of like a starting point and an end point, with the web kind of branching out in the middle, but it's a nice home plate, a start and a finish. And so it's a lot different than some of the demos that are provided by SciNote, but we are finding this format is working really, really nicely for keeping everything in, I guess, order of what we would anticipate how the project's going to go, as well as having a targeted area that we can selectively click on what experiment or what stage of that project that we're interested in looking at its progress. So it is important, I think, just because I trust that everybody's going to want to kind of have that hub of like, what animals were used, what were the protocols involved, what was the timeline. So when I open that task of our animal information, you can see under the notes I've listed our approved protocol. One of my favorite things too about SciNote is that I can actually link from the inventory the actual animals that are utilized in this project. And that's shown right here. So these are our animals that are assigned to that task flow. And then the animal information doesn't have a protocol necessarily, but in other downstream tasks, we would certainly link a protocol. So speaking of inventories, let me just take you there really quickly. So we have quite a few animals in the lab, but it's also more and more the case these days that we're getting a collection of exotic brains. So I thought it was really important to create an inventory for kind of our in-house laboratory rats that are in our vivarium, and then have other inventories for all of our tissue, as well as this new project that we're working on, the wild rats, because they are trapped locally, but obviously they are not housed in any of our facilities. I felt like that was important to have another inventory. And of course, our standard chemical inventory and antibody inventory. So this I really have enjoyed getting able to put some students on these projects, because if you have a student user, they can upload to the inventory as well. And you can just see we have a list of your standard antibodies and your other chemicals and reagents that you might need. And then I've also added these different columns to see where did they come from, where are they stored, are they hazardous, just basic things that might be known by a few, but it's important that it's accessible by all. So let me go back to our lab rats as well, because one thing that SciNote has helped us just in the nature of creating an electronic notebook for our purpose here in an undergraduate research setting is that we are now standardizing the animals that we receive from vendors. So our vendor here is often in Vigo, and they will ship us, clean, healthy animals, but are otherwise not tagged or identified in any way. And so here we can create a standardized nomenclature so that I know what year and what animal that is, and you can also link it to different projects. So this is something that I think most labs are already doing, albeit in an Excel format, but it's nice to know that you can integrate this directly into your electronic notebook. And again, notice this assigned task or tab. This is basically saying that this animal is already assigned to a task flow, so I can directly click on this at any time and see what that animal or the tissue from that animal is being, how it's being utilized in our hands here in the lab. And then the last thing I'll just run through really quickly is the protocols. So here you can see all of our active protocols. You can also see who published it. So this is really helpful with timelines and capturing kind of a snapshot of when that protocol was created, who created it, and who would be the best person to verify whether or not that is the most up-to-date protocol that you're using. And again, you can link these to different tasks. Again, that's the column here. And so, for example, if we're looking at an IHC protocol, I can also tag some of my reagents, so I know exactly what reagent the student doesn't see that it's sodium citrate, because there can be a few different sodium citrates in the lab depending on which salt you're using. This way I can directly link that item to this protocol and there's no questions left unturned. Also, I can fill in any notes here and include supplies as well as the steps that are needed at any time. So because I created this, you'll see an edit. If I did not create this, I would just be able to view it. So that also helps kind of with the fidelity of the protocols, that things are not easily changed. It's not just a click of a button and it changes. This is a really nice kind of secure way of creating a timeline of how things are done in the lab. So I will say that it does take a concerted effort of a team. You need at least 1 or 2 dedicated people to think about how your lab operates and think about ways that are best to disseminate information in a way that makes sense not only to those who are intimately involved in the day-to-day, but those who are coming maybe for a few weeks to shadow or a semester. And so that will take some time to sit and think about how to best implement that. But it's not, months and months of work. If you know how to enter things into Excel, if you know how to type up a protocol in Word, it's really just learning the system and keeping up with it, right? Anytime that you're utilizing a system and you step away from it from a little while, you come back and you're a little rusty, but it's pretty easy to pick back up, and there's updates that are always coming to kind of help make it more user friendly. So we've really enjoyed having a way to utilize what's been set up by SciNote's platform and adapting kind of how our lab works to what it can do. And I will say that their team has also been extremely receptive to real roadblocks where I was finding, I have to have the function this capability, and I can't. And Vid and other team members were very receptive to hearing our concerns and our needs, and I've definitely noticed some of those adaptations coming in future updates. So I'm happy to address specific questions, or if I've missed anything, please let me know. I'm happy to go back over, but this is just kind of our years worth of figuring out how to best integrate multiple years of research into one platform. So thanks again for your time.
Thank you, Jacy, so much for this presentation. We have first questions already rolling in, and we'll start with the debate now. But before we get into that, just that we have, so that the panelists get a bit more understanding in what your needs are, we're gonna launch now a poll. So if you wouldn't mind just clicking, you should see it on your screen. If you wouldn't mind clicking, that would be great. And maybe this is also the first question for you guys, Kelly and Jacy. What was, so we have here, so the question is, what are your lab's main struggles you're hoping to solve with the ELN? So if you could go back in time when you were deciding, what was your, kind of, even if it's not on this list, but what were like the main reasons that that made you look for an ELN?
Yeah, the tracking of the protocols, things change, chemicals are discontinued, and we may get a new product or change the dilution, those details. So tracking inventories, protocols for specific projects. I think we're all interested in anything to enhance our efficiency and productivity. But those were the two main reasons that we were searching for something to help.
All right, great. Jacy, anything to add?
I mean, definitely keeping track of everyone's work and progress, moving from our composition notebooks to having an actual secure database that was digitized was a huge concern and need. And I would say too, just to emphasize what, or reiterate what Kelly had said, sometimes we have an antibody that's working great, and we have everything worked out, and then they just discontinue the antibody, and you have to kind of start over, or find a different vendor. And I think there's been a number of examples of that happening in the last few years. But I can tell you again, from my experience just setting up our inventory, I've noticed that I can actually create a place where I have the actual lot number. And because I'm tagging that back to the original task, I know exactly what antibody that was. So if we ever have problems in the future, I don't have any question about what was actually used in the lab, which is really, really important for, kind of publishing and just continuing that knowledge between students.
And just to add something, that because I'm at an undergraduate institution, I was becoming more sensitive about, I wanted students to see a role model and to see how science could be done. And I felt like I needed to step up my game in order to share that so the students can see how information needs to be categorized and treated a little bit more formally than we were able to do in the past with our old-fashioned notebooks. I wanted some model for them to see as well.
Yeah, and I was happy to help with that. I was actually really excited when we were thinking about integrating an ELN to our lab, because coming from my PhD experience, I was an authorized user of controlled substances, which is a big deal, right? Like, you can't just mess around with controlled substances, and everything is regulated to the T. And so having that kind of background, and that was what I was comfortable with, I was really excited to get to integrate more of this regulated kind of streamlined accountability, like Kelly has mentioned, with, even if it's an antibody, a completely unregulated, molecule, at least we know exactly where it came from, how much we had, when it was used, so that we're as transparent as possible.
Mm-hmm. And I really like to both kind of alluded to this. So like Jace, you mentioned like the linearity of let's say how traditionally we think of science, or even like you know, how paper notebook forces you to you know, think linearly about experiments. And it's something that when we conceptualized SciNote, this is something that bothered us as well. Like that it's every, you know, it's, as you've shown, like experiments get very complex and very messy very quickly, which is hard to describe in a linear fashion. So I want to maybe, because I've seen some questions going in this direction as well. So I wanted to pick your brains on how do you, how did you come up with, you know, the complex structure? Was it like, was it difficult to switch to this? Kelly, you were mentioning that you were forced to conceptualize. So if you can speak about this process, because I think this is perhaps the most daunting task when you're used to working in a certain way, and then you have this system that allows for this complexity. How do you, you know, make sense of things and bring order?
Right, I think just reiterating and kind of going back and verbalizing what we were doing. I mean, we had always conceptualized it, but it's just translating it to this format. So we know what we do. It's just thinking about how to enter it in and the trajectory. So that added an interesting layer, and I love seeing it depicted this way because I usually didn't see when we write our, even when we write up our manuscripts, it seems, because it's all on a page, it seems to be a little bit more linear. But yeah, so just our protocol dictated this process. It was just a different way of visualizing it. And then, as I said, Jacy actually put the, not the pen to the paper, but the format to the program.
Yeah. So there's definitely some legwork that needs to be done on both sides. Maybe to Jacy's words, like we do put a lot of effort from our side trying to understand the processes, the processes from our customers. All of our support staff used to work at the bench. So we try to be as helpful as we can, but do expect some level of, you know, figuring things out and mapping your existing processes to the software. Yeah, so it's just the nature of everybody does science a bit differently, and it's finding these commonalities and finding the things that yeah, your department needs to adjust. So
you're
yeah.
right, and finding the key denominators, common factors, like starting with the animals or protocols for antibodies, they're going to be similar or the same regardless of the experiment. So finding those common protocols that are central, and then things that are more specific to the experiments. Again, we're all doing this, it's just visualizing it in a little bit different way.
Yeah, absolutely. Then I wanted to talk a little bit about you. So Kelly, you were mentioning 21 colleagues, 9 organizations. Then we were talking about collaboration during COVID. Obviously, digital tools help here immensely. But maybe if you can talk about a little bit how this was to set up, and to refer to one of the questions, by you know, bringing people in, sharing this information, you are sharing potentially trade secrets, copyrights. How do you prevent data leaks? How do you control who sees what? Right.
So a little clarification. So we're only sharing in our lab right now. So we have we have multiple users, but it's for my lab, it's myself, Jacy, our postdoctoral fellow, our postbac, and then a research technician. So there are 4 people who can add, change a protocol. Students can pull down protocols as they're doing things, but they're not going to add information. So that's all within the confines of within our own laboratory. in a recent grant I submitted, which was a multi-PI grant, and we haven't done this, I incorporated the idea of having of each PI being a user on a different SciNote platform, so that we would have access to each other's information that would be specific for that grant. So I have not really implemented that because I've not received funding for that. So I think that if you did the multi-PI version, and Clement, you may speak to this, maybe this is, I think that this is possible, because I think I mentioned it to Vid when I was writing the proposal. So it'd be information that would be specific to your lab. Generally, again, I'm more educational and collaborative, so we don't have too many secrets, but I understand there may be government, grants and such where things would need to be confidential. So you can control who can see, who can add information, but I would envision maybe having different setups if you had multi-PI situations, so that everything that you're doing, they probably wouldn't want, it would just be too complicated to have everything. So just be specific for that grant. That's the idea that I had. And Clement, I don't know how well that represents what would be possible with a multi-PI.
Yeah, how we typically handle this, like you as the publisher of the data with it, well, it's not really public, but if you are the person who is uploading data to SciNote, you have complete control over who sees that data. So, and you can change this as the time goes on. So even if, let's say somebody's working on a project with you and they change their position, or they should no longer have the privileges of seeing this, you can really fine tune this, per project essentially, or also on the organization level. So that there are built-in features to manage this, but still, you can share, you have the power to share this. So you should be still very vigilant with whom you share, similarly as having paper notebook. If you give it to somebody, they'll get the data. So with the power of, instant share essentially comes also the responsibility. So yeah, that's definitely a valid point there. And couple of questions, like I see a couple of similar questions, and I have the same. How do you handle the data capture at the bench? Like you were mentioning using SciNote for all sorts of let's say things, but how do you bring SciNote to the bench, or do you still use paper notebooks? How does this the actual use of the person in the lab look like?
Jacy, do you want to? That's a good question.
Yeah, absolutely. So I would probably best describe it as a hybrid. So we are still very tangible. Pen and paper is one of our preferred kind of initial data capture methods. But having said that, our hope and goal is to have a simultaneous iPad or some other tablet format that has the protocol pulled up, that the students can follow along, so that if they're doing an IHC plate, they'll have the protocol that's loaded onto SciNote to guide their work. But, to be honest, a lot of our work is done at the microscope. It's kind of difficult to get a laptop next to your microscope. A lot of us are not blessed with tons and tons of space. So sometimes the pen and paper is the best way to go. However, what I will say is that even though a lot of our initial data capturing is either through worksheets that we've created, or images that we're snapping at the microscope, all of that gets uploaded into our SciNote project, either through a scanner. So I'll scan those data sheets that the students are writing all of those results initially captured. So I'll upload through a scanner, or if they're taking images, that gets uploaded into our results section. So it's kind of a nice, almost backup method. So we do still keep binders of the paper data collection. However, it's all been digitized and kind of localized into this single platform.
Yes, and I do really love having the students just write down manually. I think there's still some value to that. They see that, and we have that extra layer of supervision when they bring the data to Jacy or a postback to make sure it's appropriate. They may check coefficient of variation or something to make sure it's valid.
And if there's, so let's say, for example, we're running a behavioral experiment and we're just recording times or latencies or rewards accepted, things like that, that can all go into either a table format. It can be adapted in some easy way to upload. But it's important too that in those behavioral experiments, a lot of times observations, really critical observations are made in like little kind of side notes. And so those are brought to me directly. The goal is to have either daily or weekly kind of powwows with the students so that I have everything that they've collected that day or that week. Depending on the season, whether we're in an academic semester or the summer, it might be a little bit more or less successful in doing that. However, the point is that those notes that are captured in the midst of the actual behavioral experiment or the trial, that can all be uploaded by typing into the notes section of each of our tasks under the results tab. So again, I have another way to ensure that it's not just on a side margin of someone's notebook, that it's actually been integrated into the appropriate spot.
Okay, great. We did some of that because we get asked that a lot, like, you know, do we have to throw out all the paper notebooks if we're switching to your system? And it's, to be honest, it's something that ultimately should be the goal, but realistically, there is a transition period, and most labs lack the interactive interface at the bench to be, you know, to be really able to transition completely to be a paperless organization. We did some of the research, you know, what's the ultimate interface for the lab? Is it tablet computers? Is it laptops? Is it, you know, virtual reality glasses, Alexa? So what is it? You know, the answer is quite boring. Like, for the time being, tablet seems to work the best by far. So something that we are playing now with is to include tablets as part, so lab grade tablets as part of SciNote subscription. So if this is interesting for any of you listeners there, let us know, and we can try to figure something out. But yeah, ultimately, on the long run, we want to include the interface for the use of SciNote at the bench as part of the subscription. We are developing our app as well, so that you know, the experience of using SciNote at the bench should be as seamless as possible, and that it should take all the strengths of paper notebook and, you know, add to those the power of smartness and connectivity at the bench. So yeah, I think these are really important questions, and yeah, maybe not straightforward to understand when you start with an ELN project. A couple of questions are hinting towards what is how did you motivate people? And I'm going to be I'm going to be maybe quite direct, but often with from the industry we hear, oh, we just tell people that that's how they'll do it, and they do it. We don't hear this as often from academics. You know, academics stereotypically have more free mind, a lot more freedom in how they do their experiments, so, which is the right thing. But you know, some things you just have to come together and agree on a common system. So how was that? Did you have like some people that were really against these changes? Students, how did you motivate them? How did you set up yeah, this organization?
Yeah, so again, we're very small. So it's our leadership in the lab is 4 of us, I guess, who aren't undergraduate students. So as I mentioned, just during COVID, we had some initial Zoom meetings about what do you think about this ELN, and then met with VID, and everyone was very supportive that they would give it a try. So the students, we want to incorporate it, some sessions. I think Jacy's going to do a session for our summer students this summer. They know that we're using it just so that they can see exactly what this is and why we value it. So yeah, so I guess we're different than a large lab and in an industry, or even a larger PI that has several postdocs and lots of graduate students. It does seem to be, as Clement was saying, you can regulate who can upload the information, who can see the information. But I think it's important, at the end of the day, PIs are responsible for the accuracy and integrity of the science. So once we decide how to improve that, hopefully we can be persuasive and everyone will be on board about how to do that. So I'm just speaking with a very small lab, and others of you out there may have more experience. And I can't imagine that there'd be too much pushback, but I don't know. I'm not out there with a lot of people with individual, agendas and such. So I guess that's a personnel management kind of question, but a good one.
Yeah. Okay, so we are getting a lot more questions and we need to slowly start wrapping up. I think we answered, so there are quite a few technical questions that we can get back to you and describe in detail how things are done in SciNote. But maybe just to wrap things up, thinking more about maybe more philosophical questions. How the book also that you mentioned, right? There's so much things happening in science. We see so many positive results being published, but very little negative results or null results. Digitalization plays a role in this, helping everything more transparent and everything. We talked about just before we started the webinar how incentives definitely play a role, and incentives, the way the system is, the scientific system is designed, is to incentivize basically publish or perish. But if we take incentives out, if we don't think as incentives as part of the problem that we'll solve, what are things that we can do as scientists, how does implementing digital systems impact our science? What would you say as sort of end remarks? What should we do as scientists to make sure that our science is more credible, more relevant?
Yeah, I mean, all of that. For me, it's being more accountable and for what we're doing, and having that transparency, and trying to make sure that what we are publishing is truly, it's accurate. We all think that it is, but all of these details are important. And I'm fortunate to be in the teaching aspect of research, and it's the process that is so important. And just today or yesterday, we realized that some students, a protocol, there was a mistake. And I was a little bit in a bad mood yesterday because I realized we're going to probably lose those data. But then I reminded myself that it's the learning process, so we can still go through the protocol and talk about why this was important. We learn, so every time something does not go right and I get over being moody about it, I learn a whole lot. So I wish the incentives could be switched to what did you learn from your research instead of how many citations you have. And as some of us are getting older, I know I am, I'm just more focused on, we have a lot of problems that need to be solved. And so we need to be less effective, but less influenced by, well, it's easy to say and idealistic, by a number of publications and whether or not we get these grants. We have to have resources to do research. But being more accountable, teaching students to do this in the right way, and to realize that we call it a negative result, but a result that doesn't support our hypothesis is still just as interesting and sometimes more interesting. So if we can at least, I'm trying to reinforce that, and I really always have with the students. But once you get to a tenure evaluation or a grant application, that's less relevant right now. But I think we all need to keep our eye on the ball of just answering the questions in the best way we can and learning from that. And it's still good science. When something doesn't work out, there's a reason it didn't work out. So our views change as we age out in science. But the book made me really look at things differently, and I think it'll be great for the students to read. I'm looking forward to our little book club to discuss that.
Jacy, how do you see the future of science unfolding? Like, what do you expect?
Saw her eyebrow go up.
What do you expect? What do you foresee as like main changes in academic research going forward?
Gosh, it's not a loaded question at all. No, I think that we need to embrace the digital age, right? So we are already, and this sounds so silly, but one of the things that I was starting to have trouble with on the day-to-day with our students was that sometimes I couldn't read their handwriting because they don't write much. They type I'm a millennial. I'm not that much older than them. However, penmanship was important, but these are important things that if you can't read what's in the notebook, that's not helpful, right? So now that we're in a very compatible digital age, we really need to be moving in that direction. And because we can upload an Excel form or a Word document, the barriers are limited in that manner. So it's even if someone, we were talking about how you motivate people or how do you get people to adapt. Even if you're not fully comfortable with using the system, you have ways to contribute to it as long as you have 1 or 2 team members that are well-versed in the platform. Going back to your question about the future, I try not to be a cynic, but I do worry a lot about, what are our goals now as scientists? Are we asking the right questions for the right reasons? Are we funding projects that ultimately will make a difference, or are they just inflating egos? And I'm still a postdoctoral scholar, so I have my whole scientific career ahead of me, but I want to be involved in meaningful research. I want to ask questions that answer holes in our knowledge that have been either ignored or overlooked for a long time. And I think to some extent, the research culture in the United States is trying to do that. We see certain initiatives, we see certain calls for proposals that target some of these overlooked areas. But, when it comes down to it, science is a business and it's money. And I hope that we don't lose our vision of pursuing knowledge and truth rather than prestige and personal financial success. But without getting too touchy-feely, that's kind of where I am in my outlook. So pursuit of truth. And honestly, Stuart Ritchie's book touches on that as well. We are scientists, we are troubleshooters, we answer questions for the pursuit of truth and nothing else.
And just as an example of something that has affected us both lately, I mentioned the wild rat studies. How many studies have been done? How many rats have been used in research in mice? And laboratory rats, that's been my business. And just to get out there in the wild and look at things that are important to us, like stress hormone levels, their spleen sizes in the wild are 4 times bigger. There's our lab rats stress hormone levels are maybe 10%. And it makes me wonder, are our lab rats even real rats? Or is this industry for these mice and rats to be used? So we need to branch out. We're all about and should be very excited about diversity with our personnel, but we also need diversity with our protocols and species that we're looking at. And but someone who wanted to study wild rats probably couldn't get tenure because it's just too hard. So we think about those things. Are we using the right models? And what does the industry support? So we need to be diverse across the board, I think, to have more representative and relevant answers. So those are some of our struggles.
Well, Kelly, Jacy, thank you so much for this. I can't help but, feel the enthusiasm from both of you, and the genuine curiosity, and the scientists that we meet, like, are really the representation of this curiosity. And that there are some, let's say, black sheep in the herd, but vast majority of scientists that we meet are just genuinely curious and would like to do proper science. So yeah, we will do our part with SciNote to make that as seamless as possible and as transparent as possible. And yeah, we'll fight other battles like financing of this and changing the incentives in the future, probably with some webinars on this topic as well. So I know Kelly and Jacy would have a whole topic on this as well. So maybe another time. But thank you so much for sharing this.
Thank you.
It's been really nice for me personally. I've learned a lot. I'm seeing here comments from our team. They're really excited. They really liked it. And thank you to all of you participants. We got a lot of comments, a lot of questions. I think this is like the record for the webinars, for our webinars. So we'll get back to all of you. It might take a day or so, but yeah, we'll try to reach everybody and share whatever we can. So
thank you for providing some new tools to help us visualize our research and try to do a better job. It's a process for all of us. So
all right. Thank you, everybody, and see you on the next webinar.
Great. Thanks for having us.